So hello and welcome friends, and this is a Quioma session from Chambers, and today we will be having a rapid review some important topics from blood physiology. This is a live session and we have students also who will be answering, will be contributing in this quiz. So let us begin that. Let me share the screen to jot down some points if I have to. Is everyone ready? Yeah. Everyone is ready? Okay. So can you tell me what are the factors which are dependent on gamma carboxylation? Which factors are dependent on gamma carboxylation? First tell me the number. How many proteins? So we have 6 proteins, a total of 6 proteins which are responsible for which will be, we will need gamma carboxylation of these factors, that is 2, 7, 9, 10 CS. How to remember? 2, 7, 9, 10 CS. So factor 2, factor 7, factor 9, factor 10, protein C, protein S. Okay. Okay, very nice answer. Can you tell us if there is deficiency of vitamin K, what would be the first symptom? What will you appreciate first? You'll be appreciating hemorrhage. So you'll have, what is the deficiency symptom? That is hemorrhage. By the way, what is the source of vitamin K? Food source. The food source will be leafy green vegetables, and this much is okay. What is difference between serum and plasma? Difference between serum and plasma. Beautiful answer. But which factors? Very nice answer. But which factors? So serum has no fibrinogen. Serum has no fibrinogen. Serum has absent which factors? Factor 2, factor 5, factor 8. Factor 2, factor 5, factor 8. And what else is special in serum? If it is S, then S should be serotonin. So serum has serotonin very much high. Serum has very much high serotonin. Okay. What is antithrombin 3? What is antithrombin 3? Yes, kindly contribute. So antithrombin 3 is basically a protease inhibitor, isn't it? It is a protease inhibitor of which system? Intrinsic system or extrinsic system? Is it protease inhibitor of intrinsic system or extrinsic system? So it is protease inhibitor of intrinsic coagulation system. Okay. So can you tell us what are the sequences of fetal erythropoiesis? So at birth, who is doing erythropoiesis? Who is doing at birth? Which organ? Beautiful answer. At birth, liver. At birth, liver. Liver is doing your erythropoiesis from 6 weeks to birth, isn't it? Okay. So in adults, who is doing the erythropoiesis? Bone marrow is doing the erythropoiesis in adults. Okay. What is the form of hemoglobin in adults? HbA1. So what are the constituents of HbA1? Alpha 2 and beta 2 chains. HbA1 is made up of alpha 2 and beta 2 chains. But fetal hemoglobin, HbF, it is made up of 2 alpha chains and 2 gamma chains. Very nice. 2 alpha chains and 2 gamma chains. Okay. Next question is, erythrocyte, platelets, eosinophil, basophil, neutrophil, monocytes. Which lineage are they coming from? Which stem cell? Which common stem cell lineage? Beautiful answer, myeloid stem cell. That's a very nice answer. So what is lymphoid stem cell giving rise to? Lymphoid stem cell is giving rise to plasma cell, T helper cell, and T cytotoxic cell. Right? What is leukoerythroblastic reaction? Leukoerythroblastic reaction. What is leukoerythroblastic reaction? Leukoerythroblastic reaction means when there is shifting towards the immature RBCs, isn't it? Leukoblastic and erythroblastic. Okay. So which condition has leukoerythroblastic reaction? Which condition? Myelofibrosis. Myelofibrosis. Okay. Can you tell us which cells are the cells of acute inflammation? Acute inflammation are neutrophils. Neutrophils are the cells of acute inflammation. How many lobe nucleus? How many lobe nucleus for neutrophil? That's a beautiful answer. Multi-lobe nucleus. So which enzymes are present in neutrophils? In the specific granules, which enzymes? We have alkaline phosphatase, we have collagenase, we have lysozyme, we have lactoferrin, we have, there is an enzyme, specific enzyme, that is LAP. What is LAP? Leukocyte alkaline phosphatase. Leukocyte alkaline phosphatase. Does the neutrophil contain azurophilic granules? Yes or no? Does the neutrophil contain azurophilic granules? Yeah, it contains azurophilic granules. It contains azurophilic granules. So what is present in azurophilic granules? It contains proteinases. So azurophilic granules, what does it contain? It contains proteinases. It contains myeloperoxidases. It contains beta glucuronidase. It contains acid phosphatase. Okay. So that's right. Okay. Dole bodies are seen in Dole bodies. Dole bodies are seen in dash. Dole bodies. Dole bodies are seen in neutrophils. Dole bodies are seen in neutrophils. What are Dole bodies? Dole bodies means light blue inclusions. These are light blue inclusions in the periphery. That is known as Dole bodies. Okay. Can you tell us some agents which induce chemotaxis in neutrophil? Can you tell us some chemotaxins, chemotactic agents which attract the neutrophils? So we have C5a, interleukin 8, leukotriene B4, and 5-HAT. What is 5-HAT or HETE? 5-HAT means this 5-HAT is actually making leukotriene. Okay, leukotriene. So these are all what? Chemotactic agents. C5a, LTB4, 5-HET. And also, do you remember kallikrein from the coagulation pathway? Kallikrein is also a chemotactic agent. Can you tell me one name of a protein present on the bacteria which is acting as a chemotactic for neutrophil? It tells the neutrophil, come, come, baby, come. Can you tell me? So the name of the protein is N-formylmethionine. N-formylmethionine is basically a bacterial protein which is attracting what? The bacterial protein which is doing a chemotaxis, attracting the neutrophils. Okay. If I say a word left shift, what does it mean? Left shift in neutrophils. Do we remember from our lectures of neutrophils? Left shift in neutrophils. What is the meaning? If something is shifting towards left, like in the leukoerythroblastic reaction, what was the meaning of that? Yes, that's a beautiful answer. It is in infectious state. So we mean by this concept, we mean we have bands. We have more bands. We have more immature neutrophils, isn't it, in the circulation? Or we have metamyelocytes, isn't it? So you see in it is CML. CML is chronic myelogenous leukemia and inflammation or some bacterial infection. Yeah, that's very right answer. So can you tell us HCO3 chloride antipotar? It is present in which cell? It is present in which cell? Chloride HCO3 antipotar. Beautiful answer. RBC is present in the RBC. Can you tell us what is the lifespan in healthy adults? RBC lifespan 120 days. Very nice answer. What is the lifespan in units? What is the lifespan in units? Units is 60 to 90 days. In units, the lifespan is 60 to 90 days. 90 days, sorry. 60 to 90 days. Okay, very nice answer. Can you tell us what is reticulocyte? Reticulocyte. Reticulocyte is equal to immature RBC, isn't it? Immature RBC. What is difference between anisocytosis and poikilocytosis? What is difference between anisocytosis and poikilocytosis? Aniso means there is size variation in RBC. But what is poikilocytosis? Poikilocytosis means varying shapes. The shapes are variable. So size and iso, poikilo shapes. Okay, easy. That's very nice. Okay, can you tell us which receptor binds to GP1B? Which receptor binds to GP1B in the platelets? Von Willebrand factor. That's a very nice answer. So which receptor binds with GP2B3A? GP2B3A? That's the beautiful answer. It's fibrinogen. That's a very beautiful answer. It's fibrinogen. Can you tell me the name of the protein which is stimulating the proliferation of thrombocytes? Which protein stimulates the proliferation thrombocytosis or the megakaryocytosis? Thrombopoietin. 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 Okay, thrombopoietin. So if I want to classify von Willebrand factor, fibrinogen, platelet factor 4, and fibronectin, under which granule? Which granule will be having these all? Von Willebrand factor, fibrinogen, PF4, fibronectin. It's a beautiful answer. It's alpha granule. Alpha granules will have this. Okay. Okay. I have a cell and it's having a kidney-shaped nucleus. The name is dash. I have a WBC. It has a kidney-shaped nucleus. The cell is dash. It's monocyte. Monocyte. That's a beautiful answer. Okay. I have Kupffer cell in liver. I have histiocytes in skin. I have Langerhans cells in skin. I have osteoclasts in bone. I have microglia in my CNS. Can you tell me what are these actually? Beautiful answer. They are tissue macrophages. Exactly. They are tissue macrophages. Okay. That's a very nice answer. So I have a helminthic infection. I have an helminthic infection. Okay. And I also have allergic reaction. Which cells will be coming? Which cells from the WBC is eosinophil? Very nice answer. Why called eosinophil? Why called eosinophil? Which stain do they give me in the H&E stain? Yeah, because of the color. So which stain? Which color? That's some more often. Yeah. Pink, very nice answer. So can you tell me which enzyme can it produce? Which enzymes can it produce? Eosinophil. Eosinophil can produce what? It can produce peroxidase. It can produce peroxidase. It can produce eosinophil cationic protein. It can produce eosinophil derived neurotoxin. It can produce histaminase. It can produce histaminase. It can produce major basic protein. What is MBP? Major basic protein. What is the function of MBP? Related with microbiology, parasitology. MBP is major basic protein. What does it do? It is a helminthotoxin. It is a helminthotoxin. It is a helminthotoxin. It kills the helminths, your Ascaris lumbricoides and all that. Okay. Can you tell me what is the cause of eosinophilia? I had taught you a mnemonic. Can you tell me the causes of eosinophilia? Where does the eosinophils get increased? Pac-Man, that's a beautiful answer, Pac-Man. So what does Pac-Man mean? Pac-Man means parasitic infection, asthma, chronic adrenal insufficiency, myeloproliferative disorders. A stand for allergic processes, N stand for neoplasia. So which neoplasia? Which type of neoplasia? Hodgkin's or non-Hodgkin's? Which lymphoma? Hodgkin's or non-Hodgkin's? It was a Hodgkin's lymphoma. It was a Hodgkin's lymphoma. Very nice answer. So E for which disease? Eosinophilic granulomatosis with polyangiitis. Eosinophilic granulomatosis with polyangiitis. That's a very nice answer. So can we know about the okay, I tell you. Do we know where does the mast cells increase? Mastocytosis. Where does the mastocytosis increase? Okay, I give you a clue. This is a skin, and there is mastocytosis. Yeah. So which place? Which mutation? c-KIT mutation. The gene is c-KIT. So c-KIT mutation leads to. No, there is a connection between the two things. Okay. So what will happen if I have a c-KIT mutation? So what I will be having? If I have mastocytosis, I will have more histamine, isn't it? I will have more histamine. What are the effects of histamine? Histamine does hypotension. Histamine can cause, if in the GIT, it will cause peptic ulcer disease. What else? Itching, pruritus, isn't it? Itching, it can do itching. What else? It can cause vasodilation. So if you have vasodilation, then you will have flushing, redness. Okay, very nice answer. So can you tell us something? Can you tell us something about where does the aspirin inhibits dash? Aspirin is an inhibitor of dash. Aspirin is an inhibitor of dash enzyme. That's a cyclooxygenase inhibitor. So it will lead to less production of which component? Thromboxane A2. That's a beautiful answer. Okay, I tell you the names of some drugs and you have to tell me where do we use them. Okay, what is the mechanism of action? I have clopidogrel, I have ticagrelor, and I have pasugrel. I have ticlopidine. They will inhibit dash. Beautiful answer. They will inhibit the expression of GP2B3 by ADP induced. Okay, by inhibiting the ADP induced expression. I have abciximab, tirofiban, aptifebatide. It inhibits dash. Exactly, they inhibit the GP2B3A. So GP2B3A directly inhibited by abciximab, tirofiban, and aptiflixatide. Okay, aptiflixatide. But aptifebatide, not fixatide, aptifebatide. But if I have to indirectly inhibit the GP2B3A receptors, so I will be using clopidogrel or ticagrelor or ticlopidine. Yeah. So can you tell us something about if I give desmopressin? If I have a desmopressin, what will it inhibit? Sorry, what will it induce? What is the role of desmopressin in coagulation? Desmopressin. I have taught you desmopressin, isn't it? So desmopressin is basically producing more what? Von Willebrand factor and factor 8. It increases von Willebrand factor and factor 8. which place who is producing von Willebrand factor from the endothelial cells. I'm talking here about the endothelial cells and also the factor 8. Yeah. Okay, can you tell us something about can you tell us a disease where von Willebrand factor is deficient? Deficiency of von Willebrand factor is dash disease. Von Willebrand disease, that's a very nice answer. So if I have a deficiency of GP2B3A, deficiency of GP2B3A, Clansman's thrombasthenia, that's a beautiful answer. I have deficiency of GP1B. Bernard-Celia syndrome, Bernard-Celia. Okay, Bernard-Celia. Do we remember the joke on Bernard-Celia? Yeah. Okay, can you tell us, I have a drug in my hand and the name of the drug is low molecular weight heparin. Which factor will it inhibit in the coagulation system? LMWH. LMWH. Which factor will it inhibit? Beautiful answer. 2A, thrombin. Thrombin. Can you tell us direct thrombin inhibitors? Can you tell us the name of some direct thrombin inhibitors, some drugs? So we have argatroban, we have dabigatran, we have bivalirudin, isn't it? Very nice. So what are dalteparin? What is dalteparin? What is dalteparin? Dalteparin is a LMWH. It is an anticoagulant. Which factor is it inhibiting? Is it inhibiting factor 2A or 10A? It inhibits the factor 10A activated form. I have fondaparinux. Fondaparinux. It inhibits dash factor. Beautiful answer, 10A. Very nice answer. Can you tell us deficiency of factor 8 leads to which hemophilia? Deficiency of factor 8 leads to hemophilia A. Beautiful answer. Deficiency of factor 9 leads to hemophilia B. Very nice answer. Deficiency of factor 11 leads to deficiency of hemophilia C. Beautiful answer. Is hemophilia A is what inheritance pattern? X-linked recessive. Hemophilia B is which link pattern? X-linked recessive again. But hemophilia C is autosomal recessive. That's a beautiful answer. I want to increase the degradation of fibrin mesh. What I will use? I want to increase the degradation of fibrin mesh. Which drugs will I be using? Which thrombolytics? I will be using altiplase, retiplase, tinactiplase, and streptokinase. Can you tell me who is producing streptokinase? Which bacteria? Streptococcus. Okay. Streptokinase, streptococcus. Can we know about bradykinin? Can you tell us some functions of bradykinin? What does bradykinin do? I taught you PPV. What is PPV? Permeability, pain, vasodilation. Permeability, pain, vasodilation. I want to convert this reaction HMWK to bradykinin. What I will be using? I want to convert HMWK to high molecular weight kininogen to bradykinin. Beautiful answer. Kallikrein. Okay. So intrinsic pathway starts with dash factor. Intrinsic pathway starts with dash factor. Intrinsic pathway starts with factor 12. What is the name of other pathway? What is the other name of intrinsic pathway? That's a contact activation pathway. So can you tell us who is stimulating the factor 12? Who is activating the factor 12 to 12A? That is collagen. That is if the platelets are already activated. That is if we have basement membrane exposure. So which extrinsic pathway starts with which factor? Extrinsic pathway starts with this factor 7. That's a very nice answer. So what is the other name of extrinsic pathway? We actually say it is a tissue factor pathway, isn't it? It's a tissue factor pathway. That's a very nice answer. I have a disease associated with C1 esterase inhibitor deficiency. What is the name of the disease in which I have C1 esterase deficiency? And that deficiency, actually C1 esterase was inhibiting my 12A and 11A. So if I have a deficiency, I will be having edema. So what is the name? Hereditary angioedema. Hereditary angioedema. Can you tell me a drug which inactivates bradykinin? Name of the drugs inhibiting bradykinin, that is ACE inhibitors. What is ACE inhibitors? Sorry, ACE, not ACE inhibitors, it's ACE. It's ACE. ACE means angiotensin converting enzyme. Okay, that is inhibiting the, that is actually inactivating the bradykinin. Okay, who activates bradykinin? Kallikrein. Who inhibits bradykinin? ACE. Okay, clear? Very nice answer. So can you tell us which factor is having the shortest half-life? Which factor is having the shortest half-life? Factor 7. Which is having the longest half-life? Very nice answer. 2. That's a beautiful answer. That's a beautiful answer. Which form of vitamin K is active? Reduced form or oxidized form? Reduced form is active. Reduced form of vitamin K is active. Which enzyme can I use to make the reduced form? Which enzyme? I want to activate the vitamin K in the liver. Which enzyme will I use? Epoxide reductase. Epoxide reductase. That's a very nice answer. If I have a gamma glutamyl carboxylase enzyme deficiency, or I have a impaired enzyme activity, which factors will be inactivated? Which factors will be inactivated? If I have a vitamin K active form deficiency, or if I have an enzyme, which factors? 2, 7, 9, 10, and don't forget C and S. C and S are anticoagulants. Your 2, 7, 9, 10 are clotting factors. Okay. So if someone is having already, he has taken warfarin, or if he has taken anticoagulants and his all factors have been inactivated, what would be the remedy for this person? What will I transfuse in this person? I will be giving a fresh frozen plasma. I will be doing a fresh frozen plasma if someone is having warfarin toxicity. Okay, and also I will give vitamin K with it. Okay. Oh, can you tell me the place where warfarin inhibits? What is the enzyme inhibited by warfarin? Epoxide reductase. That's a beautiful answer. So we have got into the, I hope that we have enjoyed this session. That's actually a very rapid revision of our blood physiology, because mostly the questions which are asked in entrance exams are actually more from the pathological point of view. But just as a quick revision of blood physiology, we had this Quioma session. I hope you enjoyed this session. If you have not subscribed to my channel, please subscribe. And thank you very much. Have a nice day. Bye-bye.